same DNA sequence. However, they have to perform very, very different functions. And so that means that we need to have different protein repertoires to give each cell its correct fate and identity. And so this image is quite beautiful to me because we're starting with an embryo where basically everything looks the same. And through the course of development, we end up with this beautiful salamander with many different kinds of cells that perform distinct functions. And this has to be carefully controlled at many stages to be able to result in this profound number of cell types. And the first step in regulating gene expression is the conversion of information from DNA into RNA. And this is done by a really elegant set of enzymes known as RNA polymerases. And today, this will be the focus of my talk, is understanding RNA polymerase regulation. The RNA polymerase that you will hear about today is called RNA polymerase II. It's been known for over 50 years to be the most important RNA polymerase for transcribing protein coding genes. And you would think that with over 50 years of studies, we know everything about it, but there's a lot that we still don't understand about its regulation, particularly in its native environment, which is in the nucleus. So here we have an image of a starfish nucleus, and it's been stained for chromatin. So all these fibers are the chromatin, and you can see that it's quite dense and compact. And I like to give this analogy to kind of give you guys a context for how compact the DNA is within the nucleus. So each of our nuclei contains about 2 meters of DNA. So if we were to take each chromosome and stretch them out end to end, it would be about 2 meters in length. And this has to fit into a size of a few microns in diameter to fit into the nucleus. And this would be essentially the same thing as taking a fishing line and stretching it from Melbourne to Port Campbell, and then trying to compact that into a soccer ball. And it's quite a dramatic challenge, and I'm glad that I don't have to do this. And at the same time, we need to keep the chromatin in the DNA quite organized so that we don't introduce breaks and other things. What I find quite remarkable is that in this very, very dense environment, RNA polymerases actually can find the right gene promoters and begin transcribing to make an RNA. On the other hand, in addition to compaction, transcription can be quite aggressive towards chromatin. It can result in the loss of nucleosomes, and this can lead to transcription then initiating in the wrong places in the genome. And so there really has to be a careful coordination between keeping the chromatin compact and accessible so that transcription can happen, but not having a case where the chromatin information is lost. And so our lab is really interested in understanding how these two processes are connected in order to give us very, very specific transcriptional products. So today, I'm going to be focusing on how transcription is regulated during the very, very early stages. And many of you learned in your introductory biology classes or introductory biochemistry classes that RNA polymerase II is recruited to gene promoters. And I think most of us left those classes with the impression that if RNA polymerase made it to a promoter and began to synthesize an RNA, we didn't need to really worry about anything downstream of that. However, there are many steps that happen during early transcription that can still regulate whether a transcript is made. So RNA polymerase II is recruited to gene promoters, and it requires over 70 polypeptides to actually form a complex that then can open up the DNA and begin synthesizing an RNA. Once it escapes from the promoter in multicellular eukaryotes, can undergo a very important regulatory step known as promoter proximal pausing. And this is what most of today's talk will be about. Promoter proximal pausing happens within the first 20 to 100 base pairs of the gene, and it's induced both by the underlying DNA sequence, as well as protein factors, including DSIF, which is here shown in green, and this red factor that you're going to hear a lot about called negative elongation factor. I'll get into why we pause in a moment. But if we do want to synthesize an mRNA, the polymerase does need to escape from this pause state. And to do this, there's a specific kinase that can phosphorylate the polymerase, as well as these factors, DSIF and NELF. And this results in NELF being lost and additional elongation factors binding the polymerase and making a polymerase that can very quickly synthesize an mRNA. So what is this pausing thing doing? It seems to be a very, very critical checkpoint during early transcription, and it has to happen after initiation, clearly. And this checkpoint is going to decide whether the polymerase goes into this active elongation step that I just told you about, or if it terminates. And this termination step can then act as a second step to ensure whether we actually want to make that mRNA. So you might be asking yourself, why even bother having this secondary step? Initiation should control everything. But we think that there are several reasons why we have this secondary checkpoint very shortly after transcription initiation. And one of those is to help to keep chromatin open. And so we know from some very beautiful studies from the Edelman lab at Harvard Medical School that if you don't have pausing, frequently what will happen is the first nucleosome in the gene will actually encroach over the transcription start site, and this will prevent all transcription from happening on this gene. So by having a polymerase initiate, transcribe, and pause, that prevents that nucleosome repositioning. We also know that pausing is really, really important whenever a rapid change needs to be made to a transcriptional program. It was initially discovered as being a very important way of regulating heat shock response genes. It's involved in immune responses, and viruses like HIV actually use this to regulate their transcription once they're in our cells. One of my most favorite examples is actually from differentiation. And so a study from a number of years ago from Karen Edelman's lab showed quite beautifully how pausing was really, really important for cells to differentiate. And in this experiment, what they did was they took mouse embryonic stem cells and they fed them serum. And when they were given serum, they start to differentiate into a lineage-specific line. And so what you can see here in a control cell line is that upon serum induction, the cells become ruffled, indicating that they've started to differentiate, and they start expressing a cell type specific transcription factor. In cells where pausing has been ablated by knocking out this null factor, the cells remain rounded and they don't express this cell type specific factor. Numerous other studies have happened after this one where people have really precisely looked at the different stages of development where an ELF is needed. It's really important for actually inducing these stage-specific transcriptional programs. So I'm a biochemist, I'm a structural biologist, and my interests really have gone to how is this pausing process actually working at the molecular level and how is it regulated? And there are actually a number of ways that pausing is regulated. One is at the sequence level. So the DNA sequence actually can encode pausing behavior. And our lab has done a lot of work in this area that I'm not going to talk about today, but I would be happy to discuss with anybody offline. We also know the chromatin structure of a gene can really influence pausing, as well as the protein factors that happen to bind to the pause polymerase. And so today I'm going to tell you about work that we've done to really try to understand at the molecular level how these protein factors are inducing and stabilizing paused conformations of RNA polymerase II. And so we're going to have three main actors for today. The first is DSIF, and it's a protein factor that's most widely conserved out of all transcription elongation factors. It's found in bacteria. It's called Nussagian in bacteria. And it's both important for pausing as well as for appropriate elongation. The other actor that we'll be talking a lot about today is called negative elongation factor, or NELF. And this protein is mainly found in multicellular eukaryotes, and its main role is only in pausing. And the last factor that we'll be discussing at the second half of the talk is a resistance factor that has activities both in pausing and elongation, but we don't really know what its molecular identity is. And I think today I'll be able to tell you what that is. So NELF is a 4-subunit protein complex. As I mentioned in the previous slide, it's mostly found in multicellular organisms, but not in plants. And it's really important for having very focused pauses at the early part of genes. Other organisms that don't have NELF can pause, but you tend to have more polymerases dispersed across the early part of the gene body. And back when I was a postdoc with Patrick Kramer, I solved some of the first cryo-EM structures of NELF bound to RNA polymerase II. And from those structures, we really thought that NELF was functioning to prevent nucleotide addition. And that's how it was functioning as a pausing factor. And today I'm going to tell you about how that view changed over the course of time from our initial structures to now. And so back in 2018, when we were first trying to understand how pausing works at the molecular level, we needed to form a very homogeneous, structurally stable complex. And so we picked a pause that we know is really strong. And this pause comes from HIV. As I mentioned before, HIV uses this to regulate its own transcription. And this pause is one of the strongest that we're aware of at the sequence level. And so using this pause sequence, we could assemble RNA polymerase on a DNA and an RNA scaffold with pausing factors D, and NELF, and then we could purify it using size exclusion chromatography and show that everything bound quite nicely. And so we were able to solve about a 3 angstrom structure using cryo-EM, and it revealed many different aspects of NELF biology. One of the main findings that we had is that when NELF was bound to the polymerase, it made an active site conformation that didn't allow for nucleotide incorporation. And there were some other features that we noticed that really seemed to indicate that NELF was blocking all productive transcription elongation. And so I'm going to show you what polymerase is usually doing during nucleotide incorporation, because it's going to become very relevant for the rest of the talk. So typically, when RNA polymerase is transcribing along DNA, it's going to cycle between 2 active site states. And so the first active site state we're going to call pre-translocated. Here you're looking at the template strand DNA and the nascent RNA here in red. And what you can notice is that all the bases are base paired. If the polymerase translocates from this pre-translocated state into a post-translocated state, what you'll notice now is there's an opening here where an NTP can come in and base pair with our DNA. So polymerase is typically cycling between this pre and this post-translocated state thousands of times as it crosses a gene. And in our NELF structure, we actually see a conformation that's very different from these two. So we're going to now visualize this using a movie. So we start with a pre-translocated state. Polymerase is going to translocate into a post-translocated state. And what you'll see now is that we have a DNA base that now can base pair with an incoming nucleotide. Once base paired, this nucleotide here in orange can be synthesized into this nascent RNA. And now we're back into a pre-translocated state. And so now the polymerase can translocate again and go into a post-translocated state, and again do this thousands of times to be able to synthesize the RNA. In our NELF structure, what we see is actually a hybrid of these two states, where the DNA is in a pre-translocated state and the RNA is in a post-translocated state. And we refer to this configuration as a tilted state. So what you can notice here is there's no space for an NTP to base pair with a DNA. And so we can't actually synthesize a longer RNA at this point. We think that the whole RNA-DNA hybrid would have to retranslocate back to this pre-translocated position, and then the polymerase would try again to move it so we could get in the next DNA base. So when we saw this structure, we saw that all of our active sites had this tilted conformation. And so this is really what led to this idea that NELF, when it's bound to the polymerase, really completely prevents the polymerase from elongating and incorporating in new nucleotides. And so we were fine with this at the time. It made a lot of sense. But this view got challenged a few years later when someone in our field published some cell biology results that differed. And so these results are coming from a ChIP-seq experiment performed by the Charlotteford lab. And in our field, one of the favorite experiments that a lot of people like to do is to take their favorite transcription factor and add on some kind of degron tag and look at how that affects transcriptional activity. So when they remove the factor, what happens? In this particular experiment, they're removing a factor called SPT6. You don't need to know what that is, but it's a really important transcriptional elongation factor. So we're first going to look at these ChIP-seq tracks before SPT6 has been lost. And what they see is that when you look at NELF, it's very, very highly enriched in the promoter proximal region, as we would expect for a pausing factor. And we see that this SPT6 factor is throughout the gene body, as well as RNA polymerase. We also see that polymerase is quite strongly accumulated in this promoter proximal pause peak. So this is in the standard condition before we've depleted SPT6. Once SPT6 is depleted, what we notice is there's no SPT6 in the gene body, which is how the degron should work. But we see that now NELF, in addition to being in this original promoter proximal pause peak, is dispersed across the entire gene body. And this came to us as a bit of a surprise because we really thought that NELF was preventing transcriptional elongation. And so this presented 2 different hypotheses to us about NELF. One could be that in this particular genetic background, NELF was coming now on and off the polymerase, and this is why you were able to detect it in the gene body. The other hypothesis is that maybe NELF was able to adopt a conformation that would actually allow it to travel on the polymerase in an elongating state, and that it wasn't always acting as this braking factor that was preventing the polymerase from moving forward. So we wanted to test these hypotheses. This was the the idea that would it be possible even for NELF to engage with an actively transcribing polymerase? And so this was work that was done by Bonnie Sue in the lab, a really amazing graduate student who was able to take a very brave approach to address this question. And so what Bonnie decided to do was she wanted to know, can I actually take an actively transcribing reaction and use it for cryo-EM studies? So I first want to walk you through what it is that we're doing here, and then go into the actual work. So here, we're again looking at transcriptional pausing, and we use a gel-based assay where we can take RNA polymerase that's highly purified and put it onto a DNA RNA scaffold where the RNA is fluorescently labeled. And then we give the polymerase nucleotides, and then it can begin to extend this RNA. And so you can see over time, we get this fully extended product, and we have these other products that correspond to pausing positions. What you can notice here is that when you include DSIF and NELF, the polymerase is slower. It takes it more time to reach these fully elongated products. And we see that this pausing position is also enhanced. So a lot of you are structural biologists. You know that in structural biology, we usually want to go for the most homogeneous, pure sample that we can possibly go for. And Bonnie wanted to answer this question about NELF, instead going the opposite direction and going for a heterogeneous sample. And so she wanted to take this 1 minute time point of transcription where we see multiple products and actually visualize it and say, what is NELF doing in this context? Do we see only NELF associated with the complexes that are associated with this pause position? And so she took this 1 minute time point, she purified the complex, and then looked at the different RNAs that were associated with it. And she could see again that it was very, very heterogeneous. And she put this onto cryo-EM grids and hoped that we would be able to classify something out of it. And indeed, she was able to classify out 2 states with NELF bound. And this was a surprise because previously we only saw this one state, which we call the pause state. And she saw a brand new state, which here I'm going to call the poise state, and we'll be discussing for this part of the talk. So these structures were both solved at about 3 angstrom resolution. Again, the active sites are going closer to about 2.5 angstrom. And what you'll notice here, if you look carefully, is that NELF has really changed its position about 40 to 50 angstrom on the polymerase. And this is a result of 2 of its modules moving. I'll make this a little bit easier to see by removing the density and now doing a morph where we're going from the pause state, which was what we originally saw back in 2018, to this new state, which we call the poise state. So you're starting here in this pause state. Here we have NELF. What you'll see is that this portion of NELF called BE goes close to this part of the polymerase called the downstream DNA, and this AC module rotates back on the polymerase. I'll play this movie one more time. So you can see that there's quite a dramatic shift. It's adopting new molecular contacts on the polymerase to go from this pause state into a poise state. So both of these states were very equally occupied. We had about the same number of particles in both conformations. And so we wanted to start asking, is polymerase behaving the same or differently now that we have this poised NELF conformation? And so here again, the conformations are quite dramatically different from each other. And the first thing we wanted to look at was the active site. And I showed you a little while ago that when NELF is in this pause state, the active site's in the tilted conformation, it can't add nucleotides. And so this is the 2018 density where we only see this tilted conformation. And here we have these actively transcribing polymerases that we're visualizing. And so we first looked at the paused state, and what we saw is that we actually got a mixture of active sites. So instead of just getting one active site state, we have this tilted state coming back, which is great. But we also see an actively elongating state called the post-translocated state. And this is with the paused NELF conformation. We next wanted to look at the poise state, which is our new state that we hadn't previously observed. And here the active site was primarily in this post-translocated state where it can actually incorporate nucleotides. So this was pretty exciting because it indicated to us that when NELF goes into this poise state, we could actually have a polymerase that could actually translocate and could incorporate nucleotides. The next thing we wanted to look at was a competition with a rescue factor. So I had these points on some of the previous slides, but I haven't really discussed them. So one of the other things that we noticed back in 2018 is that when NELF binds polymerase, it prevents a reactivation factor from binding. And sometimes that's really important when you're in a pause state. You need this reactivation factor, which is called transcription factor 2S, to bind and help you overcome that state. So I'm going to show you what TF2S does in a slightly different context, and then we're going to go back to our data. So one of the ways that TF2S can help polymerase overcome tricky sequences is in this context in chromatin. And so when RNA polymerase is transcribing through chromatin, it's quite a challenging substrate for it, and it will frequently stall in the chromatin context. So this is a nucleosome, and what you're going to see in a moment is that Pol II stops, and that's because it's become too challenging for it. And what it will do is actually move backwards along the DNA when it hits this challenging location, and the RNA needs to go somewhere. And this RNA actually needs to get backtracked through a secondary funnel called the pore in the funnel region. And this puts the polymerase in this backtrack state where it's not able to transcribe. To get out of it, you need this yellow factor TF2S, which just came in and clipped the RNA, or actually helped the polymerase clip the RNA by stabilizing a second metal ion in the active site. And so this will then allow the polymerase to resume transcription elongation and make it through this challenging substrate. And so pausing, we have a similar event going on. The polymerase goes into an active site conformation that's incompatible for nucleotide conformation or nucleotide addition, and TF2S can help resolve this. So we first wanted to investigate at a modeling level, could TF2S even engage with a NELF-bound Pol II? And so we're first going to look at the pause state and do an overlay where we look at NELF binding polymerase, and then we're going to add in TF2S. And so here's NELF. It's covering this pore and funnel region. And when we overlay TF2S, I hope you can appreciate that TF2S and NELF are clashing. And so it's not possible for TF2S and NELF to bind to this polymerase at the same time. When we perform the same modeling experiment now with NELF in its new conformation or in the poise state, we see that NELF can nicely be accommodated here, and that when we overlay TF2S, it's nicely able to go into the polymerase and it doesn't clash with NELF. So this was cool, but we wanted to say from a biochemical level if this was also possible. And so another thing that we have with this particular DNA template that we've been using for our pausing assays is that we have this backtrack species. And so I showed you a little while ago that TF2S can help overcome this backtrack species. And biochemically, what that looks like is when we add TF2S, it results in the resolution of this backtrack species when we get pausing. So if you look at our real data over here, when we add TF2S, we basically get rid of the backtrack species and only have the pausing species. Similarly, if we only add DSIF or NELF on their own, we only have a pause species and no backtracking. And if we do this experiment now with both DSIF and NELF present, what you'll see is that we don't fully resolve the backtrack species like we do in the other cases, but it does go down compared to the sample without TF2S. So this suggested to us that there might be some fraction of molecules where TF2S was actually able to access them. So Bonnie decided that she wanted to go and see, using cryo-EM, whether TF2S could be accommodated on DSIF and NELF and what conformation it would sit in. And so when she performed this experiment, what she saw is that NELF was only in the poise state, and that in this poise state, TF2S could be very nicely accommodated. And she didn't find any particles where NELF was in a pause state and TF2S was there. And this really confirmed our modeling experiments that the two would not be able to bind the polymerase at the same time. So this really changed our view of NELF just being this roadblock stopping factor. It seems like it can bind the polymerase when you're actually able to actively elongate, and it can allow rescue, but it does require this remodeling of NELF and to enable both of those properties. So the last part of this section, I just want to tell you a little bit about trying to understand pausing in general. And this doesn't have to do just with the poised and the pause state. We know that both NELF and DSIF are really, really needed to pause the polymerase. And unfortunately, in the structures I showed you today and in the structures that we determined in 2018, we haven't been able to see the direct interface between NELF and DSIF. And then so in 2018, our way of going about this to try to understand how they might actually collaborate with each other to induce pausing was to use cross-linking mass spec, where we have primary amines becoming cross-linked with each other, and then we can map what amino acids are interacting. And when we did this experiment, what we saw is that one subunit of NELF called NELF-A, which is here shown in orange, extended across the surface of polymerase. We call this region a tentacle. And these pink spots indicate where the polymerase and NELF-A were cross-linking. At that time, to confirm that this interface was actually important, I made a very brutal truncation where I chopped off over 300 amino acids to remove the tentacle. And when you do this, what you can see is that pausing is no longer as efficient, and the polymerase is able to make the full-length product. This wasn't a very satisfying answer of how NELF is inducing pausing, because we've really just removed a gigantic portion of the protein. And so since this time, our world has changed immensely in many ways. And one of the ways that it's changed is that we now have AlphaFold available. And back in 2018, we had some densities on the polymerase that we couldn't assign what they were. And so we had to kind of leave things where they were. And same thing in this structure that I've shown you now, we had additional densities that we weren't quite sure what they could be. But AlphaFold has really helped us to illuminate what it could be. And so we've done some work with the labs of Lucas Farman and Karen Adelman and Johannes Walter at Harvard Medical School. This has really been driven by Johannes Walter's Predictomes platform that's publicly available if any of you are curious. In these experiments, what we're doing is doing pairwise AlphaFold screens, where you take your favorite transcription factors, RNA polymerase subunits, and basically fold them against each other, and then generate these huge matrices where you can look at how things might be associating. And so each of these squares indicate whether you have a low or high confidence interaction. And we got excited about this, particularly for a NELF-A interaction with an RNA polymerase subunit known as RPV2. And so this was predicted to be a really, really high confidence interaction. And so when we first looked at NELF-A folding on its own, we could assess the quality of the folding using these PLDT scores. And so this part of the fold for NELF-A on its own has very high confidence. This is what we see very nicely in our cryo-EM data. And here is the tentacle region, which you can tell has a pretty terrible PLDT score. There's nothing very high confidence about it. However, when we co-fold NELF-A with this RNA polymerase subunit called RPV2, what we could see is that there's this region in the tentacle that suddenly has a very, very high scoring PLDT score that gives us confidence that there's probably a real interaction there. So we went and we looked at where this interaction was predicted to be. And we actually had density on the surface of the polymerase that we previously couldn't model. And this density could very nicely accommodate about 10 amino acids coming from this NELF-A tentacle. So we wanted to confirm that this region was indeed critical for NELF's association with polymerase and for pausing. And so Bonnie went and mutated 4 residues this time instead of cutting off a huge amount of the protein, and was able to nicely show that by just substituting out these 4 residues that make this interaction with this subunit called RPV2 of polymerase, we could basically completely overcome pausing. And so it really showed us in a much more refined way what additional surfaces are very, very important for the pausing phenotype. So over the course of the last 10 or 15 years, Cryo-EM has really allowed us to now visualize multiple factors on RNA polymerase too. And this particular region where we see this NELF-A tentacle binding to Pol II called the RPV2 protrusion is also known now to bind multiple factors at different stages of transcription. It's also used by an initiation factor called TF2F. It binds exactly along this protrusion area. And additional elongation complexes where there's a factor called LEA1 or the superelongation complex with L2, and then elongin with its ELO-A subunit. And so what this shows us is this one binding surface on the polymerase is used by very, very distinct factors, and it helps to demarcate the specific stages of transcription. So putting this all together, this study showed multiple things. One is that NELF's conformation really will affect what its role is in pausing, and it can actually go into a state that actually allows for elongation. It's not always preventing nucleotide addition. And we think of NELF associating with the polymerase quite early on in transcription, and we think that it can cycle between these poised and pause states until it reaches a pause sequence where it can then go into this pause conformation. In the event that polymerase gets backtracked or tilted or other things that need to get corrected, if NELF is able to enter into this poised state, TF2S can help out. And this can allow the polymerase then to resume transcription. And eventually, the pause will be released with these factors, spt6, PAF, and NELF, and this will allow the polymerase to go on. And so this work was really benefited by multiple technological enhancements. So by using our actual biochemical reaction and visualizing it, we're able to actually trap different conformational states of NELF. And further, having the ability to do AlphaFold screens has made our work much easier in identifying densities that we previously weren't able to assign. So this is the summary for the first half of the talk. Now I'm going to tell you another story where we are trying to understand one last factor that's involved in transcriptional pausing. And this factor has appeared in the literature for nearly 20 years now as a resistance factor. And this stems from a observation that in order to recapitulate promoter proximal pausing in a test tube, we have to use conditions that are very, very distant from what you would actually expect in a cell. And so this work has been led by an extremely talented postdoc in our lab, Roberto, and he's been on the hunt to figure out what this resistance factor might be. So something that I haven't told you all, but is very critical, is that in all of the biochemical assays that I've been showing you so far, we've been doing these with extremely low nucleotide concentrations. These are in the tens of micromolar of nucleotides, and this is to be able to facilitate really strong pause detection. In cells, our nucleotide concentrations are on the millimolar concentration level, and in the field, this has led to this hypothesis that something's missing from our reconstituted approaches. We have these beautiful purified proteins, but we're unable to get the same behaviors that we would expect in cells. And so this has led to the hypothesis that we have a missing factor, and the factor that I'm going to tell you about today is called a resistance factor, and that name will become clear of why it's called that in a moment. So one protein factor that's been proposed to act as this additional potential pausing factor is GDOWN1. This protein regulates polymerase during mitosis. It doesn't seem to be bound to DNA-bound polymerase. And so this really dampened our enthusiasm about this protein potentially being the possible resistance factor. Another factor came up more recently that we got a lot more excited about, and I hope I can convince you today that we think is probably the real resistance factor. And it's a protein called ELOF1. And the reason we got excited about it is that it's a very, very important protein in transcription elongation. Everybody who studies yeast transcription has known for quite some time that it's very important for just general transcription elongation behavior. In the human system, it's been a bit more forgotten, and the counterpart in human is ELOF1, not ELF1. And in the past 5 or 6 years, a number of papers have come out to show that it's very important for transcription-coupled DNA repair. This is an event where when polymerase encounters a lesion in DNA, it acts as a sensor, and this can lead to DNA damage repair. And ELOF seems to be very, very critical for that. It also was recently shown to have a role during B cell development. So antibodies need to generate additional mutations to be able to have high specificity for their targets. And it seems like it's very important in this role during transcriptional elongation in generating somatic hypermutation. From these studies, one of the observations that the authors of these various groups had is that ELOF seems to have a strong correlation with pausing. And it seems to be very important in pull downs. When you do mass spec, you see that DSIF and NELF are no longer as nicely associated with the polymerase without ELOF. All of these studies didn't care too much about promoter proximal pausing. They were more worried about these other important biological phenomena. And so we were curious, could ELOF actually be this resistance factor that's needed for us to actually fully recapitulate promoter proximal pausing in a test tube? And so one of the first things that Roberto wanted to do was to first confirm that ELOF is actually in the promoter proximal region of genes. And so to do this, he got a cell line from the lab of David Schatz, where ELOF has been C-terminally tagged with an HA tag. And this enables us to actually pull it out in ChIP-seq. I should note that ELOF is extremely tiny. It's about 10 kilodaltons. And it's very, very structured and not the easiest thing to tag. So using this cell line, Roberto was able to show that ELOF is very much enriched in the transcription start site of genes. And this is similar to the occupancy of RNA polymerase II. And so this was really encouraging because at least they gave us an indication that ELOF might be present in promoter proximal pause complexes. The next thing that Roberto wanted to do was to see if ELOF has a role in pausing. And so he went to his traditional gel-based experiments. And these, again, are performed at very low nucleotide concentrations. And it's the exact same sequence that you were looking at in the previous slides. And so here, what you can see is that when we add ELOF to our pause complexes, it really has very minimal effects on promoter proximal pausing. These are extremely short transcripts. The polymerase is only going for 7 nucleotides. We're doing this all at low nucleotide concentration. So we were curious, are we missing something just based on this assay? We need something where we can actually look at a much longer transcript. And so Roberto has developed a really cool in vitro transcription assay that we refer to as a cell-free genomics approach, where we can actually look at transcription on a library of DNA sequences. And these sequences currently are about 300 base pairs in length. And we can take time points after adding our factors and then isolate the RNAs directly and use these for sequencing. And this enables us to really look at a lot of different sequences. We don't have to run sequencing gels, which is traditionally how you would look at these longer RNA transcripts. And you can identify what's going on with the polymerase. And so Roberto decided to do this kind of experiment using physiological nucleotide concentration, so we're about 1 millimolar nucleotides, and then assess, do we see any differences in our polymerase activity when we add in ELOF? And what was really exciting is that he sees that when you have just polymerase basically at 1 millimolar nucleotides, it's fully transcribed these DNAs in this library within 30 seconds to a minute. Similarly, when we only have DSIF and NELF, you can see that the polymerase very rapidly completes transcription. And we see that it's much slower now when we have in this ELOF factor. And so this is a physiological concentration, and we're having a much, much slower rate of elongation. And so this was extremely exciting to us because it gave us the indication that, yes, maybe indeed ELOF is a factor that is making the pause equivalent to what we're seeing in cells. The next thing that Roberto needed to do was actually to see if this is the so-called resistance factor. And I haven't really told you where the resistance factor name comes from, but I'm going to do that now. And so resistance factor, this name comes from a phenomenon that the Price lab notices that the resistance factor prevented this initiation protein called TF2F from binding Pol II. So TF2F is here shown in purple. It's really important for transcription initiation, but it's also one of the only initiation factors that can actually go on an elongating RNA polymerase II. And what they found is that whatever this resistance factor was, it would prevent TF2F from actually binding and activating the polymerase. And so we know now why this is probably the case. TF2F binds polymerase in a very similar location as both DSIF and NELF. I showed you previously that the tentacle part of NELF overlaps with TF2F, but there is also a direct overlap between DSIF and TF2F. And so what we knew is that the resistance factor actually makes this DSIF-NELF complex completely impenetrable to TF2F. And I'll show you what I mean by that in a minute with some data. So we can assess TF2F's effect on polymerase again using this gel-based assay. And so here we have polymerase on its own. We see that it's pausing quite nicely. If we have both DSIF and NELF, we see that the pause is much stronger and there's not an elongated product. If you add TF2F to the polymerase, it actually enhances its transcription activity. As you can see here, where we have more extended RNA product. And so in the same context, if we add TF2F to our DSIF-NELF reactions, what we can see is that polymerase is no longer pausing as efficiently, but it's also not reaching this fully stimulatory capability that we have with TF2F. And so there's a competition going on between DSIF-NELF activity and TF2F. And lastly, here we have ELOF, and ELOF on its own really has very little effect on transcriptional activity. So I wanted to ask, if we add ELOF into this reaction with DCIF-NELF and TF2F, does it change anything, or do we kind of see the same activity? We'd expect that if ELOF is this missing resistance factor, we should get back to this pause state that we see with DCIF and NELF on their own. And so when we add in ELOF to DCIF and NELF, we see again we have a very nice pausing complex, very similar to what we started off with here. And if we have ELOF and TF2F, they're both able to transcribe quite nicely. But now if we do this experiment where we add ELOF with DCIF and NELF and TF2F, we basically see that we're staying in our pause state and we're resistant to this TF2F stimulation. And so we think this really shows that ELOF is making DCIF and NELF very resistant to TF2F function and fits this resistance factor categorization. So the last thing we wanted to do was to again go back into cells and to ask, is ELOF actually enhancing promoter proximal pausing in a cell-based assay? And so to do this, we use this same cell line that we use for ChIP-seq, which has a Degron tag on ELOF. And so this allows us to treat the cells with the small molecule D tag or control, and this results in ELOF being degraded by the proteasome. We can then assess early transcription dynamics by basically preventing new initiation. So if polymerase is strongly paused and we don't have initiation, we can basically watch the kinetics of how quickly we get decay of the pause. And if we think that ELOF is helping the pause, we would expect that without it, the polymerase will decay faster from promoter proximal pause locations. And to assess this, we're using a ChIP-seq based method. And so we're taking multiple time points, both at 5 and 20 minutes after trip-to-light addition, so after initiation has been stalled, to look at how much pause polymerase we have. And what we see is that when ELOF is degraded, we actually decrease the half-life of Pol II at pause sites quite dramatically. And what's really exciting is that we see that the genes that are most affected with polymerase occupancy are the ones where we have ELOF the most well-bound. And so this really suggests that ELOF is very important for helping to stabilize the pause, and it's really important for this phenotype in cells. So lastly, you guys know that we like to do cryo-EM, and this is a cryo-EM meeting. We did determine the cryo-EM structure of ELOF bound to DCIF and NELF on polymerase. And we see that ELOF is helping to stabilize this clamp domain of polymerase, but we don't see a direct interaction between NELF and ELOF. And so, as you probably are aware, anytime a structural biologist doesn't know exactly how something functions, we say that it's probably through an allosteric mechanism. And so we think there's some kind of allosteric conformation that ELOF is helping to stabilize in the context of these two other protein factors. So with that, I think we've been able to find that ELOF is probably this missing resistance factor. We're finally able to recapitulate pausing behavior using physiological concentrations of nucleotides. It's preventing TF2F from acting on Pol II, and we see that it's in the promoter proximal regions of genes and enhances pausing duration inside cells. And so I think all of this data together helps us to kind of finally understand at least what the base components are in eukaryotes to induce pausing. So today I hope I've shown you a bit more about how these protein players are involved in pausing. Again, we're curious about how other effects like chromatin and DNA sequence induce pausing, and those again are very active areas of work in our lab. So with that, I'd like to thank the lab. A big shout out to Roberto here for the work that I just showed you, and an undergraduate who works with him, Serena. Bonnie, who's graduated and is off working a pretty cool job in industry, our amazing cryo-EM facility here at MIT, and then Andreas Meyer and Ann-Kathrin and Elizabeth for all their help in analyzing our ChIP-seq data. And so happy to take any questions that you all might have.
Thank you, Seychelle. That was a fantastic talk and a cool, investigative biology story, which I don't think we get to hear too much from in this series. I know I have some questions, but I want to open the floor. If anyone has a question, please, you can unmute yourself or type it into the chat and I can read it out, whatever works. See if anyone has anything. Otherwise, I will start. Okay, I guess I'll go first. Since it's a cryo-EM audience, I wanted to ask about how you manage these heterogeneous data sets when you have these increasingly complex samples, like more and more factors and more states. Are you able to identify those different states in like your 2D classes, or how does that work?
Yeah, no, that's a fantastic question. So yes, we were dealing with both conformational and compositional heterogeneity, which certainly makes life way more challenging. And so we actually do most of this classification in 3D. And so we will initially use TD just to throw out obvious junk. And then after that, we go looking for which particles have NELF bound. And then from there, you can start classifying on the active site using lots of masking approaches. And so it's a very iterative approach, and every complex is a little bit different on what works. But most of this has to be done at the level of 3D.
Sure. Thank you. Any questions from anyone else? Mm, Maybe I'll just keep asking. Oh, here we got someone in the chat. Yes, were you ever able to resolve the tentacle region?
Yes, so we can resolve the tentacle region um, quite nicely. It was always sitting there in the density. We just didn't know what it was from. And you all probably have encountered this, that you sometimes just have densities that you're not quite certain about where they come from. And so we can resolve it. We just can't make the full connection for the tentacle region that goes to the main body of Nelfe, unfortunately. We have additional density there that we just can't assign right now.
Yeah, someone has a follow-up to that, so it's not an intrinsically disordered region. Question.
Well, it depends on what you want to call things. I guess it's like a depend. Yeah, I would say in the field, probably people would call it an intrinsically disordered protein, but I would call it a protein that's disordered until it has its binding partner. And so it's like once the polymerase is there, it can adopt a stable fold. But before then, it would be fall into that intrinsically disordered category.
Anyone else? Let's see. I guess I was going to ask then, I haven't really worked any RNA or DNA binding proteins, but I was wondering if you know much about what forces are holding the DNA into these complexes, like it wraps around the histones and that the RNA polymerase complex. Um, and obviously it has to be stable enough for cryo-EM, but if you know much about how that kind of side of it works.
Yeah, yeah. No, there's a lot going on here because you have like the the base pairing interactions with the DNA that help to stabilize things quite a bit. But a lot of the interactions by either in the nucleosomes or in the polymerase, you have lots of arginines, lysines that are helping to coordinate phosphate interactions, and then other interactions that might be base pair specific. Polymerase doesn't care about the sequence so much as like for the initial binding. It's more for translocation, but there's um, a very, very positively charged cleft where all of this stuff is sitting.
Right. I'll just wait a little bit longer, see if anyone else wants to jump in with a question. Otherwise, we might be finishing a little bit early today, but
No problem. And don't be shy, everyone. I'm happy to answer anything.
uh, oh, silence.
Oh no.
If not, I guess we can wrap up there. Thank you again so much for joining us and sharing this story with us today. Some very cool structures, and yeah, we'll look out for where the story goes next with Elof. Thank you.
Great. Thank you all.